Today, we're diving into a technology that sounds like it's straight out of a sci-fi movie. We're talking about something that could potentially cure cancer, help end world hunger, and yeah, even change the very course of human evolution. It's called CRISPR, and honestly, it's one of the most powerful tools humanity has ever created.
I mean, just think about that for a second. What if we could go into the very code that makes us who we are, our DNA, and fix the typos that cause devastating genetic diseases? What if we could engineer crops to survive droughts and feed our entire planet? This isn't some far-off dream. This is the world that CRISPR is starting to make possible, right now.
So what in the world is CRISPR? Okay, the full name is a bit of a mouthful, but the concept behind it is actually pretty simple. The best way to think of it is like a pair of tiny, programmable scissors for our DNA. It's like having a find and replace function for the code of life itself. But here's the wildest part. This revolutionary technology wasn't invented in some high-tech lab. Nope, it was discovered hiding in plain sight, inside some of the oldest and simplest organisms on our planet. Believe it or not, CRISPR is basically a bacterial immune system. For billions of years, bacteria have been using it to defend themselves against viruses. When a virus attacks, the bacteria grabs a little snippet of the virus's DNA and stores it in its own genome. It's like creating a genetic most wanted poster. That way, if that same virus ever shows up again, the bacteria can recognize it instantly and just destroy it. And the system scientists have harnessed has two main parts. First, you've got a protein, and the most famous one is called Cas9. This is the scissors part. It does the actual cutting. But, scissors are pretty useless if you don't know where to cut. That's where the second part comes in, something called guide RNA. Think of it as a biological GPS that you can program with an exact genetic address. It leads the scissors to the precise spot on the DNA that needs a little snip.
The whole process is just so elegant. The guide RNA zips along the DNA until it finds the exact sequence it's looking for, and then it locks on. The Cas9 protein then unwinds that little section of the DNA double helix and makes a clean cut right through both strands. And that cut is where the magic happens. The cell's own repair machinery kicks in, and scientists can either let it patch itself up, which usually disables the gene, or they can provide a new, healthy piece of DNA that the cell will use as a template, effectively fixing a mutation.
Okay, so we've got this incredible tool that can edit DNA with pinpoint accuracy. What does that actually mean for our lives? Well, it means we are standing right on the edge of a complete medical revolution. For the very first time in human history, we have a real shot at actually curing a whole list of devastating genetic diseases. Take sickle cell anemia. It's caused by a single typo in just one gene. And get this, clinical trials are already happening where doctors take a patient's own blood stem cells, use CRISPR to fix that one tiny mistake, and then put the corrected cells back in. They're essentially curing people of a lifelong disease. It's incredible.
And it's not just about inherited conditions. This quote here is from a high school student asking a biologist if you could use CRISPR to fix a gene called P53. Now, P53 is mutated in over half of all cancers. And the answer to that student's question? A huge, resounding yes. That's exactly what researchers are working on like crazy right now. But CRISPR's impact goes way beyond the hospital. Scientists are using it to create crops that can survive in a warming world, which could be a total game changer for global food security. And they're even editing the genes of mosquitoes to stop them from spreading diseases like malaria. This single technology could help us solve some of the biggest challenges our species is facing.
But of course, a tool this powerful, a tool that lets us alter the code of life itself, it forces us to ask some really profound, really difficult questions that we've never had to face before.
The biggest debate really boils down to this: therapy versus enhancement. I think most of us would agree that using CRISPR to cure a child's fatal disease is a wonderful thing. But what about using it to choose their eye color, or make them taller, or even smarter? The line between fixing a health problem and designing a person can get really blurry really fast. Where do we, as a society, draw that line?
And there's another really critical difference we have to understand: somatic versus germline editing. So somatic editing is when you fix the cells in just one person, like fixing lung cells. Those changes stay with that person and they die with them. But germline editing, well, that's different. That's when you edit the DNA of an embryo. Those changes would be permanent and inherited by all of their descendants forever. This isn't just a theory, by the way. It actually happened in 2018 when a scientist created the world's first gene-edited babies, sparking a massive global debate.
And then there's the huge question of access. The first gene therapies that are getting approved cost millions of dollars for a single patient.
So what happens if CRISPR cures are that expensive? Will we create a new kind of genetic divide in our society? A world of genetic haves and have-nots? These are massive questions, and honestly, there are no easy answers.
But just when you think you have your head wrapped around CRISPR, the story takes another incredible turn. It turns out gene editing was just the beginning.
So remember that super precise GPS system, the guide RNA? Well, scientists figured out they can get rid of the scissors and attach other tools instead. This means they can send a molecule to a specific gene not to cut it, but to do something else entirely, like just turn it on or off like a light switch, or maybe just make it glow so they can see where it is. This has opened up a whole new world of possibilities that just sounds like science fiction. I mean, scientists are using CRISPR to store entire movies in the DNA of living bacteria. They've built rapid diagnostic tools that can detect viruses in minutes. They're even figuring out how to make temporary, reversible edits to our genes. The potential is it's truly mind-bending. So, CRISPR has basically given us the power to rewrite the code of life. We can use it to heal the sick, to feed the hungry, to understand who we are on the most fundamental level. The question for us is no longer if we'll use this power, but how. And that's a conversation that all of us really need to be a part of.